Hello and welcome to part 1 of our lab skills series, working with cryopreserved cells. Brought to you by California North State University, College of Pharmacy. Before you retrieve your cells, make sure the sash of the biosafety cabinet is at the proper level to allow for ideal airflow. Sterilize the hood with 70% ethanol and have all required supplies at hand. To minimize airflow disruption, try to work in the middle of the hood space. Take care when removing your cryotube from the liquid nitrogen. Always use protective gloves and a face shield or goggles if available. Slowly pull the rack from the liquid nitrogen. The tank is kept quite full, and pulling the rack out quickly will cause the liquid nitrogen to splash and spill from the boxes. Pulling the rack out slowly allows for excess liquid nitrogen to drain back into the tank. The cryotube should be put on ice immediately to prevent melting, which will impact viability. Swirl the tube in a 37-degree water bath to melt the freezing media, approximately 1 to 1.5 minutes. Keep the cap of the tube above the water level to avoid contamination. Swirl the tube until a small ice crystal remains. Spray a Kim wipe with ethanol and wipe down the outside of the cryotube before placing it into the hood. Always remember to spray your gloved hands with ethanol before performing any cell culture activities. Begin by transferring the contents of the cryovial into a new 15 milliliter tube. Rinse the cryovial with 1 milliliter of fresh, pre-warmed cell culture media. Slowly add the media to the tube to avoid osmotic shock. Add additional warmed media up to 8 milliliters. Cap the tube tightly and label. Centrifuge the tube at 1500 times G for 5 minutes to remove any excess freezing media. Spin speed and time requirements will change with each cell type. Be sure to find the best settings for your cells. After centrifugation, you should have a well-formed pellet. Aspirate the supernatant, being sure not to disturb the cell pellet. Resuspend the pellet in 1 milliliter of media using the P1000. At this point, you can add media up to 5 ml to perform a cell count as shown in part 3 of this series. Alternatively, if you know the number of cells that was originally frozen, you can resuspend the pellet in a specific volume to achieve a desired concentration for seeding. Add media directly into the flask first, and then add the cells in. Pipette up and down to mix. Cap the vented top of your flask tightly. To ensure homogeneity, be sure to rock the flask gently in all directions. Label the flask clearly with your initials, the date, cell type, and passage number if available. Move the flask to the microscope and view the cells to ensure they are present at the intended density and homogeneity. Check multiple areas of the flask. Move the flask carefully to the incubator. Culture time and conditions will vary based on cell type. Thank you so much for watching. We hope this has been helpful. Please come back for more lab skills videos brought to you by California North State University College of Pharmacy.